Hello everyone and welcome to another class of Physiology for Engineers by Professor Gonzalez-Fernandez at Lehigh University. Today we are going to talk about the nervous impulse and how the nervous impulse is originated and then is transmitted through the axonal body to the next neuron. So there are two different processes that we need to understand in order to address this nervous impulse. The first process is the initiation of the nervous impulse that is going to happen here in the axonal body. So this is called the graded potential. And then there is another process that is going to take this graded potential that is originated here in the body and is going to conduce it or take it until the axonal terminal here where all the neurotransmitters are going to be released in order to excite or inhibit the next neuron. So this second process that we are going to look today, which is a conduction process, is going to be the action potential. Okay. So first of all, we are going to focus on the initiation or graded potential, and then we are going to move to the conduction or the action potential. So the graded potential has different characteristics. The first characteristic is that, first of all, is the initiation of the nervous impulse. And the second characteristic is that this initiation depends on ligand gated ion channels. Okay. So this ligand gated ion channels, I'm going to focus on that now. Here we have our neuron, but then there is going to be a previous neuron with an axon terminal here that is going to have some neurotransmitters. And then those neurotransmitters here in the vesicle, they are going to be liberated into the synaptic gap, and then they are going from the presynaptic neuron to the postsynaptic neuron, so presynaptic. We are going to look at all of this a bit more in depth when we talk about the chemical synapses, but it's good to start introducing the terms now. The presynaptic neuron and the postsynaptic neuron. So these neurotransmitters are going from the pre to the post, and then they are going to bind to these ligand-gated ion channels. Okay. So these binding are going to activate these ligand-gated ion channels, and they are going to allow for the exchange of ions between the extracellular space and the intracellular space according to the concentration gradient, as we have seen in the previous video, which was the membrane potential. Okay, so, and this takes us to another characteristic of this graded potential. So the third characteristic is that it can be positive or negative. So if it's positive, it's going to be depolarization, and if it's negative, it's going to be hyperpolarization, depending on the channel that we're activating. So if we are activating, for example, sodium channels, we are going to allow the entrance of sodium into the cell. So if we look again at the schematics or the graphs for the action potential, so here we'll have millivolts, and here we have time, same here, same here. And if you remember from the membrane potential video, the resting membrane potential is around -60 to -90. So let's say that this neuron is -70. Okay. So if we have this activation of this ionic channel, and we have the sodium entering into the cell due to this concentration gradient, what we are going to have is more positive charges inside the cell. So we are going to here make the membrane potential more positive, okay? So this making the cell more positive is called the depolarization of the cell membrane. But Also, we can also activate different channels. So if you remember, the neuron had not only sodium channels, but it also had potassium channels, and it also have chloride channels. Okay, so the potassium channels, they are going to follow the concentration gradient, and they are going to pump the sodium outside the cell. Therefore, they are going to pump positive charges from the cell to the accessory space, and they are going to make the cell more negative. And something similar, but in the contrary sense, happens here in the chloride channels. So the chloride is also going to go for the concentration gradient. So it's going to bring chlorides into the cell, and it's going to make the cell more negative, right? Because the chloride is negatively charged. So this process is called the hyperpolarization. So what is going to happen is that it's going to make the membrane more negative. Okay. So those are the first three characteristics. Let's continue. So now, also, the other characteristic is that the intensity, how positive or negative this gradient potential is going to be, is tunable. So we can regulate intensity so there are two ways of regulation. So it can be temporal. So it depends on how many times these channels can get activated. So they can activate more repeatedly. We are going to increase the intensity. And also it can be spatial. So there can be more than one sodium channel activated, right? So if we have another, we are going to also increase this depolarization further. Okay, so because this intensity of the changes in the membrane potential are tunable, they can also be added to each other. So this is another characteristic, it is that we can add the gradient potentials one to another in order to increase intensity, right? And this is very important because in order to start the conduction phase, to start the action potential phase, here at the trigger area, so this is the trigger area, we need around -50 to -50 millivolts, okay? In order to activate these voltage-gated channels that are going to start conducting the potential to the axon terminal. So let's say that we have here one channel, sodium channel, so that's going to increase, let's say, to -60, right? So then if we have another, we have an addition to -50, and then we are going to be able to activate the action potential phase here in the conduction phase. So again, we have different characteristics of the gradient potential, and some of these characteristics are different to the action potential, so we're going to compare each other. Most important, the gradient potential is the initiation of the nervous impulse in the neuron. Second, it depends on ligand-gated channels. So these ligand-gated ion channels are going to bind to these neurotransmitters, and they are going to activate the channels. It can be positive or negative, so it can be depolarization or hyperpolarization. And then it also, the intensity can be tuned through the activation repeatedly of the channels or through the activation of more channels. And finally, these changes can be added to each other, so these gradient potentials can be added to each other in order to have a summatory effect that is going to give us the needed voltage potential to come here to the trigger area and start action potential. So now give me some time to get rid of some of the drawings, and we'll go into the action potential section.